what I'm going to try to do is I asked Brian if I should talk about machine learning or I should talk about ecology, and he told me to talk about ecology. So if you'd rather hear about machine learning, you can blame Brian. But what I wanted to introduce you to is this kind of very rich theoretical tradition that exists in ecology. So I think, this is models, inference, and algorithms. And I think one thing that I was surprised about over the last decade is really how rich theory in ecology is, and how, at least as a physicist, and I would say as someone who's pretty well versed in systems biology, how ignorant we are of all this rich theory. So I thought what would be nice is in this primer, I kind of just laid out the key concepts, showed you some kind of very simple general results that seem, that shape a lot of intuition in ecology. And then after that, what we're going to talk about is that a lot of this ecology was actually developed thinking about trees and plants. And when we want to apply it to things like microbes, it turns out that there's some shortcomings, even though it's a wonderful, rich theory. And it's our opinion, the people who are largely shown on this slide, who are really, I come here as a representative of this collective, that we have to understand metabolism if we're really going to take all these concepts, ecological concepts, and apply them to the microbiome. So the primer is basically set up as follows. I'm going to give you some basically very high level introduction to this, to community ecology, the theoretical community ecology. And then Josh is going to take over after about 10 slides and explain to you why we think you have to integrate that with a theory of metabolism and a theory of metabolic niches. So as Brian said, please interrupt me with questions. I'm a physicist. I get really nervous if I talk for 20 minutes and no one interrupts me, because basically since I was a first-year grad student, I've been interrupted after every slide. So if you have a question, please interrupt. Please feel to interject. All right. So that's the plan of the primer. So let me start with a very, very, very high level idea of what is ecology about? What are we interested in when we want to understand the ecology of the microbiome, right? And I think there's basically a few key questions. The key questions that people are interested in are, how do ecosystems function? How is the function of an ecosystem governed by the community, the microbial community, or whatever community, the community of organisms that live there? And how do these communities respond to perturbations, right? So this could be very macroscopic things like climate change, like global warming, or it can be like, I changed my diet. How do things change? And what's interesting is, of course, that ecology is really concerned with basically how you relate these three things to each other, or community ecology, I should say. And the thing that makes this, of course, very, very difficult question in general is that these systems are very complex. They're very, very heterogeneous. And people have, of course, in an experimental setting and both in macroecology and microecology, tried to tame some of this complexity. And so I think a lot of what you see in the way ideas and things are organized is that there is essentially, different levels of complexity you can consider. So you can consider the real ecosystem with all its kind of large scale, craziness that we can't control. And then, people have then, tried to catalog this by doing field surveys. And this is what I would say something like the Human Microbiome Project or the Earth Microbiome Project is. It's just a giant field survey trying to catalog what's happening, what isn't happening. The next level of taming the complexity is that you can kind of intervene in these ecosystems. So you can say, if I perturb the ecosystem somehow, what happens? So one famous, these are things where there's a famous ecology study where someone went to a beach and just basically removed all the conchs on the beach and asked what happened to it. So just crazy stuff. And you can imagine in a microbiome, you perturb things by adding antibiotics, you do something like that, you ask how things happen. And then, of course, things start looking more and more tame if we now, instead of thinking about really real ecological systems, we try to make microcosms, right? So we try to recapitulate communities in the lab. People have done this with macroscopic ecology by basically making the most famous example of this in community ecology is something called Tillman's grassland plots, where David Tillman, who's a very famous ecologist at the University of Minnesota, basically made lots and lots of replicate ecosystems of prairie grasses and asked what happens. And finally, of course, we would like to really eventually, the goal, at least, every time I go to a microbiome startup webpage, the claim is that we're going to be able to control these complex systems in the wild. But this is basically, this kind of controlled setting is where most of the research happens. But of course, what we'd like to do is be able to think about how we translate things at these kind of very controlled settings in a lab to these realistic things, really more realistic settings. And the claim is, again, that in order to, be able to abstract from what we learn in a lab setting to realistic settings, we have to look for some general principles, right? I'm trained as a theoretical physicist. I still kind of believe in theory as opposed to data science. And so that's what, I think a lot of ecologists also believe in that. And so they've developed these kind of very general theories. And so one way people think about it.
Can I ask a quick question?
Yeah, no, please do.
My name is Paul Reginato. I'm curious about, so I just wanted to ask about the concept of function in ecology, because I think a lot of us are used to thinking about function of like a component within an evolved organism, where the organism itself is like, a unit of selection and has kind of probably a different kind of individual nature than an ecosystem does. And so I'm curious about like the meaning of function in this context. Is function just like the dynamics that happen, or is function more of like is more of a nuanced concept than just the dynamics that go on?
So it usually means more when they say function. So of course there's the dynamics, but there is this whole field of ecology called ecosystem services. So that's what it's called. It goes under that name. And there, the idea is that the ecosystem is playing some functional role. Like, for example, a prairie will filter water. So if you've ever been to northern Jersey, they restored all these kind of grasslands because the idea was that these grasslands naturally prevent erosion, or they filter hard minerals. Other thing that people often think about is, the total biomass that's produced. So that's a function that's given. So you have to remember, ecology also has a very applied setting. So if I want to manage a forest, I have to think about what properties of the forest I want to keep going. And so this ecosystem services literature very much thinks about function the same way. And the other thing I would say is the analogy with evolved organisms is that it's unclear. So if I have a pathway that functions somehow, of course the gene is evolved, but I think the gene in isolation has very little to say about the function of the whole pathway or the whole organism. So I think these kind of ideas of how you bridge scale from, say, gene to pathway to organism are basically, there's a one-to-one translation from these systems biology problems to these ecology problems, where you can just say, how do I think about species to groups of species that function at the same trophic layer to a whole ecosystem and how they produce function. So I think these are problems that are generic. Maybe Josh wants to add something to this. I don't know, I can't see him, so I don't know if he wants to or not.
No, I think that sums it up nicely.
Great, thanks.
All right. Thanks for asking. It makes me really feel better. You don't understand how much better I feel if someone asks me a question. So having said that, I think one general way people think about this that we found very appealing is this kind of idea of a meta-community framework. And the basic idea of a meta-community framework is that you kind of have these local communities, each with their local environments. So you have a kind of a community, you have a local environment that has some basic environment-specific thing. And what you have is you have migration between these communities, all right? And this is basically the idea. The idea is that there is some species pool, which basically is the sum of the species that occurs in all these meta-communities. And basically each local community can be thought of as immigration from these other species combined with kind of like dynamics that happens locally, all right? And so this is the meta-community framework. And what's really important is what are the major ecological processes that happen? And you'll see that there's a lot of analog with evolution, since it was already brought up, which is that in each local community, you basically have two sets of variables you really have to care about. You have the actual species that live there, whose population I'll describe with this kind of standard notation, NIA, which is the number of species I in meta community A or in environment A. And these 5 processes are essentially what's going to happen. The first one is what in ecology is called ecological selection. And it should be distinguished from evolutionary selection, but it's basically the idea, how fit is this organism for the environment it's growing in, right? And this GI is supposed to be essentially a growth rate. So it says, in this environment, defined by these environmental variables EA, how well do I grow? So that's called selection. The second thing major process that seems to play a major role in all this stuff is dispersal, which is this basic idea that like I might be able to migrate to new communities much easier or colonize, or I might not, how often do I colonize? And you see this dispersal is the second major process that is really important. The third thing, just like in evolution, that plays an important role is drift. And drift is essentially demographic noise. It's population level noise, right? So some places you can have a lot of noise, a lot of stochasticity, you have very stochastic environments, or you have stochastic things, and other places are very deterministic, right? And depending on how these selection and drift, just like in evolution, you can get very different regimes, you can have very different regimes in ecology. Now, often in many, many theories, people just stop there, they talk about the species, but what we're going to try to argue is that in the microbiome, you actually really have to think about the environment. So what I'm going to try, I think a lot of this talk is going to be about, and these talks are going to be about how we can't ignore the environment. And often that's a big fallacy in a lot of what we do. And so in the environment, what happens is, of course, there's some externally, this supply is essentially the external, the external environment. These are the abiotic factors that are there. If I go in the northern latitudes versus the tropics, there's just going to be different environments that are supplied. But the environment also changes because the organisms themselves go back and impact and modify the environment, right? So these are basically these 5 processes that ecology is trying to balance: selection, dispersal, drift, and environmental impact. And just like in evolution, I don't know how familiar you are with, different ecological theories basically emphasize different processes and neglect other processes, because it's very hard to treat all 5 processes on equal footing. And so basically, you kind of have niche theory, and niche theory is really dominated by thinking about selection and dispersal, right? It says that I'm really going to think about selection and dispersal, and that's what I think is the dominant thing. So this is like a high selection regime. Then, just like you have a neutral theory of evolution, you have a neutral theory of ecology, which surprisingly was only developed in the last 2 decades. And there what you do is you say selection is kind of negligible. What's dominating everything is this drift, this demographic noise.
Hey, Pankaj?
Yep.
We have a question in the chat on that point.
Yep.
Can you say more about drift? The question specifically is, what is the ecological source of drift?
Well, it's just, imagine you just have a species and it's just undergoing birth-death process, right? Because I have a finite number of organisms that live there, you're always going to have stochasticity. So in fact, neutral theory just says all organisms just undergo birth-death process with some fixed birth rate and death rate. And what you have is you just have stochastic sampling with birth-death, and that's going to give you a population structure. So it's really like the demographic process of birth and death giving rise to a distribution. It's like if you're in the systems biology literature, it's the same reason, it's the equivalent of intrinsic noise in genes. I make a protein with some average rate, and then you don't do it with, and you degrade with some average rate, there's going to be fluctuations, right? And now you can just replace the protein with species. And that's literally what neutral theory is about.
Thanks, that's clear, I think. Is there anything that we can relate to in terms of like what might differ physically across different environments that would affect rates of drift?
Rates of drift. So it's basically rates of drift.
Stochasticity or any other sort of.
Yeah, so in the basic idea is that the rates of drift are set by the total population, right? If I have huge N, then stochasticity is small. If I have small n, then stochasticity is big. It's the usual thing, right? Just the total number of organisms is setting.
Like binomial sampling or Poisson noise or that kind of idea, right?
Yes, it's exactly like that. It's basically an Ernst-Ern process. It's just like the Ernst process is literally how drift is modeled in neutral theory.
Oh, right, okay.
Thanks.
I actually had a question. Yeah, sorry, I'm Raman Ganti. I was actually just curious because I thought the selection term would have the birth.
Well, it has a birth part too, right? The mean is not independent of the variance, right? So it's just like in gene expression. I don't know what your background is, but but it's just like the same process. The mean, so the mean is usually put in the selection, and the variance is usually put in the drift of the same processes, right? The same processes have a mean rate, and that goes in the selection term, and the variance goes in the drift term. That's usually the way it's thought about.
Okay.
Right? I mean, that's just the way it's usually thought about. It's an artificial your point is right that it's an artificial separation because the same processes give rise to both the selection and the drift. The question is just whether you can neglect the variance or not.
I mean, I guess I'm used to thinking about it as maybe a master equation, and so in that case,
yeah. So just the mean moments. So if you take the first moment of the master equation, that's the selection term.
Yeah.
Right. So
yeah.
So that's what it is. All right. And yeah.
A question here. This is Luca. Pankaj, is it clear that for a given ecosystem you can identify this environment,
No.
this community? I mean, it seems that already there is a big assumption there that a sort of separation of scale assumption in here.
Only if you in lockable Terra systems there is, because basically what you do is you say that I can neglect changes in environment and I can think of it as a species only interaction thing. Here in principle, I haven't assumed any separation of scale because I'm just tracking all the environmental variables and the species environments at the same time. But maybe I'm missing what you're saying. Because the environment has a dynamics too, right? That's what this is supposed to represent abstractly.
No, but you're saying that you in this framework you think of migration between one environment to the next environment, for example. And here I'm just saying, when do you make that decision versus saying that that's just one big environment?
Oh yeah, you have to make all these choices. There's lots of choices to be made. That's all I wanted to say. And in fact, like whole theories, whole schools of ecology make different choices. And a lot of the debates, I would argue, are about what choices you make. And in the language of physics, for the two physicists I at least know are on the call, I would say this is really about which effective theory applies. But I think everyone can understand what effective theory is, right? Like depending on what you think is dominating, you might want to use a different framework, right? Neutral theory is all about drift. Niche theory is all about selection. Locke-Volterra says that environments can be largely neglected. There's a time scale separation, really. And so I can just think about species-species interactions, and the impact of the species on the environment aren't that large. That's or are very easy to observe in species-only interactions. And the real theory is, of course, this monster beast that we can't do much with most of the time. So I just want you to know that there's these processes. And so what we're going to focus on here are niche theories with some noise added on and some stochasticity and things like that.
Sorry,
and so
one more
theories,
question on the previous slide. Sorry.
Yeah.
Just in the chat, why is the diffusion constant di independent of environment A in your model?
Oh, that's just because we're writing a review and we haven't caught all the typos yet. So thank you for pointing that out. It will be fixed in the when the figure comes out.
Thanks.
So what I'm going to focus here on is on niche theory, because in microbiomes we think, again, this is what we think, and I'll show you evidence throughout the morning, that really it's environments and environmental filtering that are setting most of this stuff. And so the idea of niche theory is an old one. The most famous example of niche theory is something called the competitive exclusion principle. And the competitive exclusion principle basically says species compete for what are called niches and competitively exclude each other. And what niches means, I have not found a non-self-recursive definition of a niche. It's kind of like, you know it when you see one. It's a slot in an ecosystem that some species fills. But how you define the slot is by thinking about what species fills it. So I'm not sure there's a independent way of defining it. But the basic intuition you can get from this kind of very famous picture on the left, which was something called MacArthur's warblers. So the basic idea was that because of competitive exclusion, which was, formulated by this very famous ecologist called Hutchinson, his basic idea is that every species that lives in an ecosystem must be best at something and excludes everything else. So there was a big mystery because there were these warblers, which are two kinds of birds, red and yellow depicted here. And everyone wanted to know how come they can coexist on a single tree, right? So they live on some tree, and eventually some people observed very closely and they figured out that the red people live on the barks, the yellow people live on the yellow birds live in the tree, and so they're all best at, they actually live in their own independent niche. The niche of this thing is not the tree, it's just if I observed more closely, I would realize the yellow birds like the leaves, the niche is the leaves of the tree, and the red is the bark of the tree, and they're both best at using these different parts of the ecosystem, right? And this basic intuition basically says the number of surviving species is strictly limited by the number of limiting resources in an environment, right? It says selection dominates. What sets how many species can survive? Well, how many things any species could be best at. So the number of species is strictly limited by the number of limiting resources. And I will show you is just a mathematical statement from a class of models. And then in low dimensions, you can translate that statement into something stronger, which says each species is best at utilizing some resource. All right. And these models are all developed using this something called the consumer resource model. And this is going to pop up over and over again for the rest of the talk. And I'm going to be done in 5 minutes, so Josh has time. And the basic idea is that the consumer resource model, I now assume particular forms for the resource dynamics and the species. And what's really important is the idea of how the growth rate depends on resources. And the idea is that the growth rate basically means that all the species just consume these resources, and the whole thing is defined by these kind of consumer preferences. Let's say, how much of resource A do I want to use? How much of resource B do I want to use? And so on. And so, we can come to these niche theory vectors. You basically.
Well, hold on. Sorry, Pankaj. That last one went a little fast.
Okay, so it's going to pop over again. But the basic idea is you have consumers, you have species, and they can eat all the resources. And the resources are substitutable. So you can imagine these being different carbon sources.
Yeah.
And each bacteria can eat all these different carbon resources.
Where is the substitution in the equations?
It's because the growth rate is a sum of all the resources.
Okay.
So I can trade 2 glucose for 1 acetate. Right? It's not that all the resources. So if I had resources that were like carbon and nitrogen, I couldn't trade off a carbon source for a nitrogen source.
Okay. And then the resources are getting consumed. And what are the Ks?
The Ks are just the carrying capacity. This dynamics will change, but this thing basically says the resources in the absence of consumers.
Oh, I see. They're getting consumed, and there's also a logistic type.
There's also a logistic term. So
okay,
this is just the classic thing that describes most ecosystems.
Okay.
Most macroscopic ecosystems. It's not going to apply to bacteria.
I just, if I see an equation, I want to understand it. So.
No, I understand. and so what I'm going to do is actually skip some slides, and maybe, am I going to tell you something? Yes, I'm going to do this slide, and then I'm going to skip. So more generally in niche theory, I don't have to actually assume a form. What's really important is that this can be turned into a graphical model, a graphical thing. And so what you have is you have species grow at some rate, GIR, that's the growth rate. Then the resources have some supply vector. This is the vector that tells you how much of resource R is supplied. And then they have what's called an impact vector, which says, how does the presence of species J impact resource alpha? Right? Everyone's basically okay with these. So there's these, and so if we want to worry about steady states, there's basically what we have to do is at steady state, we know that the growth rates of all species have to equal 0, because that's what defines a steady state. And this is called a zero growth null plane, right? And these are called impact vectors and supply vectors. and this is going to be my last math slide. So we can actually now just simply derive competitive exclusion, right? In order for the species to coexist at steady state, the ZGNIs, the zero growth milk lines for all the species must intersect, right? And you can quickly convince yourself, because if there's M resources, generically, at most M species can coexist, because generically, if I have the intersection of M minus one dimensional hyperplanes, right? I need M of these to intersect, and basically, generically, that can only happen with M resources. And so this is an example with 2 resources. So with 2 resources, these are the zero growth milk lines, so green, blue, and orange. And generically, only 2 of these lines will intersect, right? And you can do that with generic surfaces. So it turns out to be a problem in random geometry of high dimensional surfaces, which is kind of a fun problem. But generically, we know that if I have a system of equations, M system of equations with M variables, right? If I can have this many solutions. So it's just a mathematical statement of competitive exclusion. That's what it says, right? So that, and of course, if I have drift and all these other things, it's going to get more complicated. So I'm going to skip all this stuff. And what I'm going to argue is that this kind of basic picture, which is dominated ecology, is inadequate for understanding microbial ecosystems. And the main reason is that they kind of assume that I can really think about resources and environment very differently. Like I can kind of separate these trophic layers. But it's not true in the microbial world, because in the microbial world, you don't eat another organism. It's not snakes eating rabbits or squirrels. It's actually, they produce and consume small molecules. And for this reason, if we really, what it means is that cross-feeding means that microbes construct their own environment, and we must understand how metabolism creates these metabolic niches that are occupied by these bacteria. And this is this book that basically has influenced me a lot. It's this book by Levins and Lewontin, my crazy Marxist comrade ecologists. And they have this kind of like very funny throwaway paragraph that I was reading 10 years ago that really forced me to think about this a lot, which is the incorporation of the organism as an active subject in its own ontogeny and the construction of its own environment leads to a complex dialectical relationship, between the triad of gene, environment, and organism. And so because you can't separate environment from organism, and metabolism is what governs that in the microbial world, in order to really apply ecological theory, you have to understand metabolism. And I think Josh is going to tell you about that.
I'll share my screen now. So we've got just not too much time here, like 12 minutes. So I might skip a little bit too.
Feel free to go a few minutes extra, Josh. We can also, we can take a 5-minute break after you're done.
Yeah, no, you have more like 20 minutes if you want.
Yeah. Okay, cool.
Yeah, you're good.
So I'll back up here. Okay. Yeah, so as Pankaj said, kind of entering into the world of microbes means that we have to kind of presumably have a better understanding of metabolism and how that affects the environment. And, I came from a background of thinking about metabolic networks more generally. And it's been useful. The field's really evolved over the last 30 years. And I'll talk a little bit about the utility of metabolic networks more generally. But the way I like to think about it, or the way that we thought about it with working with Daniel Segre, was that metabolic networks within the microbial world is not just only a useful tool, but a way to naturally bridge scales in the biosphere. And the intuition is that the same sort of carbon flow through individual cells that's responsible for making new bacteria is directly related to the carbon that's actually cycled throughout the entire biosphere. So one may be able to think about metabolism much more generically at a variety of different scales, and it seems like a very natural sort of way to bridge scales on shorter and smaller spatial scales to like larger spatial scales and larger time scales. And we use these networks, sort of different levels of different approaches to ask questions about the evolution of the biosphere and also the evolution of individual organisms. So, but just to motivate a little bit more into why, it's interesting to think about microbes engineering their own environments, comes from this one example that I wanted to highlight, where this Russian ecologist, Winogradsky, in the 19th century, basically he was the discoverer of chemoautotrophy, which is the ability for microbes to fix carbon dioxide in the environment from electron sources that are not driven by light. So what he did was he basically made a concoction of in a water column, where he basically put mud water at the bottom of a column. And he supplemented that mud with basically a carbon source and a sulfur source, packed in another layer of mud on top of that without any kind of amendment, and then a final layer of pond liquid, and like an air sort of surface. And what happens reproducibly in these microcosms are that at the beginning of the experiment, the microbes at the bottom of the tube will start degrading the carbon sources that are provided and the sulfur sources to basically completely oxidize the sugars and complex polysaccharides into carbon dioxide, and will also reduce the sulfur sources and make hydrogen sulfide gas. What happens is that the next layer of this system basically takes phototrophic or sulfur, it's called green sulfur bacteria, which basically can fix carbon dioxide using hydrogen sulfide as a reductant. So it's a direct byproduct of the metabolism of the first layer, both from the basically produced carbon dioxide and the hydrogen sulfide to start cycling those molecules to make biomass and create energy. As you go up, you get more and more diverse bacteria, but at the very top layer, you have cyanobacteria that can fix carbon dioxide, produce organic substrates, as well as oxygen, which then feed the lower layers of the sort of aerophilic zone of heterotrophic bacteria that consume both oxygen and the carbohydrates. So this happens on the time scale of months, but it's a very reproducible system. It's called a Winogradsky column. But the key point is that the microbial end products that are produced, these layers, can basically feed other microbes at various layers that use a very diverse array of energy harvesting schemes. And the carbon sources and energy sources are used to synthesize ATP and biomass in a very sort of different way. I should mention, though, that this has been a mainstay of soil science for on the order of 100 years now or more, and only recently we've developed sort of high throughput sequencing techniques to start really investigating the diversity of these communities that emerge in these systems. And what you can find is probably what you would expect if you have any experience with analyzing like human microbiome data, is that there are hundreds, if not thousands, of individual species in each one of these layers. So our understanding of how metabolism shapes these environments, it's not simply, it's most likely not as simple as this sort of low dimensional representation of, sort of energy, carbon and energy substrates that are really cycling through it. It's probably much more complicated, in terms of what mediates sort of diversity in these ecosystems. And that really, just to kind of give you a sense of like what we understand about from the microbial ecology sense, about cross-feeding more generally, is that we really have sort of two major views of what cross-feeding and syntrophy look like in microbial communities. And the first really is that kind of like what I was just suggesting, which was that the major sources of cross-feeding in microbial metabolism is really driven by energetic processes. So fixing carbon dioxide or taking an electron donor and electron scepter acceptor, and then sort of shuttling it around throughout these different diverse energy metabolisms. And this first example on the top right of this plot is acetogenesis, where you basically have a particular microbe that can oxidize short chain fatty acids like butyrate or propionate into even shorter like waste products like acetate, and will pull electrons off these molecules and reduce protons to make hydrogen gas. That hydrogen gas then is used as a reductant or electron source to fix carbon dioxide and eventually produce methane and methanogens. So this is a very classic syntrophy. And the thing that matters for this particular interaction is that thermodynamically, at that first reaction, the first energy yielding reaction from the acetogen is actually very close to thermodynamic equilibrium. So the presence of the methanogen, basically making sure the hydrogen gas is at a low partial pressure, ensures that reaction can run forward. So syntrophy in this case really means that the presence of the second organism, this methanogen, actually benefits or really I should say makes feasible the metabolism of the first. Cross-feeding, as we've sort of understood or started to understand a little bit more about how microbes have a very diverse array of cross-feeding patterns, we can understand that it's a much more generic sort of phenomenon. So it's not just driven by energy metabolism, but there are a lot of, sort of small molecules that are excreted into the environment that can be reused, including, molecules that are excreted by isogenic lines and can be reused by the same sort of genetic, or like the same microbe in the same genetic background. The most famous example of this is a E. coli that's been basically evolved in a static environment over a period of time. It diverges into basically a primary consumer and a secondary, consumer of acetate. So that's more of a similar kind of energy-driven process. But the other examples that might emerge are what they call auxotrophies. So you might have certain bacteria or classes of bacteria that are deficient in making, precursors for new cellular material like amino acids or vitamins. And that's what this bottom example kind of shows, is that you might have a cross-feeding pattern that are, sort of more in line with typical trophic, intuition that Pankaj discussed earlier, where you might have a class of bacteria that can degrade a polysaccharide, produce small molecular weight sugars. Those get fermented by other bacteria into small organic acids. But you can have these sort of more complicated interactions where the production of arginine or riboflavin, which is a vitamin, can now feed back into the other community members because some of these members are deficient in making these products. And I should say this is why a lot of microbes are hard to culture, most likely, is that when they're grown in the community, they have a lot of dependencies, and then when you try to isolate them on selective media, they're challenging to grow up. So how do we think about modeling these sort of interactions more generally, starting from our deep knowledge of biochemistry and metabolism? Can
I ask a question just to clarify, because I think a lot of people will be a little bit less familiar with, so if anyone is down on my dumb level, I guess that's the best way to put it. So my dumb question is just to clarify and make sure I have it straight. Syntrophy we can think of as these reactions are really coupled between the two different processes. And as it states there, the existence of the second one is making, or the one on the right, the methanogen. The existence of the metabolic reactions there is really making the sort of first reaction on the left there happen more frequently or be more favorable reactions. So they're coupled in a very direct way. Whereas in cross-feeding, you're producing some byproduct, and the production of that byproduct and the consumption of it might happen or it might not. It's sort of irrelevant to the initial production of it. Is that correct?
Yeah, that's my understanding of the definitions within the field at this point.
Great.
So one can imagine that leads to all sorts of questions of whether or not how could the sort of generic cross-feeding, so you can understand how the first like centrophy you would imagine evolution would select for something like this. But the question is like, well, why do you have evolution selecting for sort of more complicated cross-feeding patterns? And there's a whole theory on, sort of loss of particular metabolic portions of the genome that would lead to sort of these dependencies, just because they're sort of leaked in the environment kind of randomly. And that's called the Black Queen hypothesis. So that's still like an active area of research of really trying to understand how evolution shapes these kinds of interactions.
Cool, thanks.
Yeah. So just to kind of continue on the idea of trying to understand, not just being able to predict, these kinds of cross-feeding patterns, or more generally from like first principles, is that, we can start with this kind of understanding is that metabolism from at different scales, at certain scales is sort of quasi-universal. And when I say that, I basically mean is that microbial metabolism can be partitioned into, is typically classified based on, what the carbon source is, whether or not it's CO2 or fixed carbon like sugars, and what the energy source is. This is a typical sort of schematic of how we classify microbes based on energy metabolism. And you can see based on the Winogradsky column example, that predicts some coarse grain understanding of, what molecules get, cross-fed in these different zones in the column. But if you take another sort of approach and say, well, okay, they're all different forms of the same function within or same pathway within a microbe, which is really just a function to take carbon and make precursors that eventually go into biomass, and then also take energy and store it into high energy molecules like ATP or NADPH that can be used to synthesize compounds. So the way that metabolic structures or metabolic networks are typically structured in this case is that you have two sort of classes of pathways. One are the breakdown of nutrients and the synthesis of ATP and other high energy compounds. And then you have, and that's called catabolism. And you have another process called anabolism that takes these building blocks and these high energy compounds to synthesize macromolecules, proteins, lipids, polysaccharides, that are eventually used to synthesize new cellular material. So thinking about this sort of more generically, you can imagine that, if you have different microbes with slightly different, metabolic networks, you could imagine there might be sort of a resource allocation strategy where secreted of, in addition to consumption of particular nutrients and just energy dissipation that you would have during this process, some strategies and under certain environmental conditions might lead to excretion of, particular metabolic intermediates that are, not quite balanced with other ones. So for instance, if you have like too much nitrogen or your primary source is an amino acid and you have another carbon source, you're basically would be uptaking too much carbon for the amount of nitrogen that you actually have in the environment. So you'd probably, as a microbe, want to get rid of that excess material somehow. So the patterns for, sort of this resource allocation problem and figuring out which metabolites get excreted is actually not really particularly intuitive. So from that really, trying to address this problem, I would say the technique that came out of, started in the early 90s, to try to understand more detailed about whether or not an individual microbe in a fixed environment will secrete, certain molecules. Really, the technique that kind of emerged in the early 90s was genome scale metabolic models, or I should say constraint-based models of metabolism. And this field was really born out of the metabolic engineering world, where the goal was to take an individual microbe and understand, well, if I can manipulate different genetics in that microbe, can I shunt or funnel food into different byproducts that are of high commercial value? So that field started in the early 90s, and up around 2000, they realized that they could use it as a whole sort of cellular model of resource allocation and metabolism to make all sorts of interesting predictions on how cells are going to rewire their metabolism in order to achieve sort of optimal growth rates. And the field has actually progressed pretty extensively since then. And these all come down to basically starting with a DNA sequence or a genome of a microbe, annotating that genome, and then identifying based on the genetics that are within a particular microbe what the set of metabolic reactions are. From that set of metabolic reactions, you can represent that as a mathematical object in this basically a stoichiometry or stoichiometric matrix, where you model the individual fluxes as variables, and the stoichiometric values basically when you have this matrix and multiply by this vector of fluxes, that represents the first time derivative of the intracellular metabolites. And the real, once you have this representation, you can do all sorts of analysis with this topologically. So you can look at the connectivity, sort of generic network-like properties. You can look at modes within this network that are sort of typical extreme points that are sort of flux distributions within this network. But I'd say the most important technique that came out of this is really the advent of flux balance analysis, which simulates the actual flux distribution through these networks. And the real, sort of the things that kind of enable that technique is that an assumption that the steady state metabolites intracellularly are basically are at steady state and not changing. So this now turns this system of ordinary differential equations basically sets the left-hand side of this equation to 0. And you set up a constraint matrix that's for every single intracellular metabolite, you have a particular constraint. And the fluxes live in this space of linear constraints. I should say, in addition to just the topology of the metabolic network, there are a lot of other important sort of pieces of information that go into building these models. One is sort of the sort of constraints on the directionality of the individual fluxes. So the thermodynamics generally sets boundaries or conditions on what reactions are going to be forward or allowable to go forward or reverse. So you have to have a good understanding of the thermodynamic constraints for every single reaction in the network. And again, this is about we're talking about thousands of reactions. And I would say that we don't have reliable thermodynamic estimates for a good chunk of these. You need to have a good understanding of the boundary conditions of this network. So the actual exchange rates or maximum exchange rates within the environment. So for E. coli grown in glucose, we have a good understanding of what the maximum uptake rate of glucose is in a fixed environment, but we don't have that for every other metabolite. We also have to have a good understanding of the biomass function. So for these cells, right? So like what proportion of lipids, carbohydrates, and amino acids constitute a new cell, and it's not clear how different that is among microbes. So this all being said, I just wanted to say that even with going from genome to model is actually a pretty tricky process that requires a lot of pieces of data. And just to give you a sense of, for the E. coli MG1655 standard systems biology model, which the initial model was built in 1990, there are still models that come out. I think the most recent one being just a few years ago, that make drastic improvements on model quality. So, and it's taken 30 years of both automated and manual intervention to build high quality genome scale metabolic model for E. coli in this case. Which kind of leads into the last point on this, just more generically, is that, so using this tool to go to ask questions of ecology, you can imagine where all of the errors would kind of compound and how challenging this would actually become. So you not only need to have reliable sequencing and annotation pipelines of particular microbes, you need to have, estimates of free energies. You need to have estimates of maximum uptake rates. And once you move to sort of the dynamic process of predicting the interactions with the environment over, larger temporal and spatial scales, you need to have a good understanding of how metabolites diffuse through environments and what the kinetic constraints are on individual uptake rates for all of the possible byproducts that can come from, transported in and out of a cell. And right now, we really don't have a whole lot of measurements, for these kinds of things. And then kind of begs the question.
Real quick, I'm sorry. Do you want to take just 1 or 2 more minutes to
Yeah, so this is kind of leading into, what Pankaj will eventually talk about. And I don't think I need to go beyond this really, because I think Pankaj is going to get into this pretty in depth, is that, one question, instead of using this very high or detailed view of metabolism to predict cross-feeding interactions, specifics, is a question of whether or not that level of detail really matters for predicting generic properties of statistical properties of ecosystems. So for this, we basically decided to go the opposite route, rather than starting from this very high resolution view of predicting specific routes of metabolism, is that you can take the consumer resource model that Pankaj mentioned in the first part of the primer and add terms that enable the exchange of nutrients within the environment. And rather than, so the typical consumer resource model is this on this left, where you have the consumption of particular nutrients by individual microbes, and you just add an additional term that allows for byproduct secretion into the environment. And as Pankaj is going to show, this leads to a lot of interesting new dynamics that are fundamentally different than if there was no engineering of the environment by microbes themselves.
Thank you for having me. Thank you for sticking through all this with us. So I'm going to tell you about some work, and like much of what's good, at least in the science I've done, it's really a collective of many, many people's ideas. I'm just presenting them. In particular, my postdoc, Bobby, and my student, Wenping. Bobby I would make him give this talk, but he left science to go join the priesthood. So he's in Rome to training to become a priest. And as you can see, we've been thinking about these ideas for a really, really long time. And these are just a list of papers where some of these ideas exist. And so I think the goal of this talk, and I guess at the Broad, this is kind of a more controversial talk than I thought when I first prepared this for a physics audience. I think the goal is to try to argue that maybe we can make predictions about large scale patterns that we see across microbiomes without really necessarily having to measure every little detail. And that this is something that physics has taught us is a very powerful way of thinking about the world, and maybe it also applies to the biological world, as long as we're willing, there's nothing for free, as long as we're willing to restrict this set of quantities we want to make predictions about. So the central thing I want to start with, a very broad level thing that I want to start with, which is that microbiomes are really diverse, right? So in biology, we often like to study things in isolation. We have this kind of very reductionist paradigm where we like to think about things in isolation, a single bacteria, maybe 2 bacteria. But if you go out into the real world and you go, colonize, for example, the human microbiome, which has gotten a lot of press everywhere, any place you look, you see many, many, many microbes living together with each other, right? And this seems to be a generic property of microbial communities. And what we'd like to understand is how we can understand it, how we can model it, what can we predict about these things, and in general, like, what are the principles governing these communities? So one of the things, as we've started to do surveys of all these communities, that's really become really interesting and striking, is that there's these kind of very large scale reproducible patterns that you see across the microbial world. So, you know, one of the first large-scale surveys of microbial communities was this human microbiome project, where people just took lots of individuals and did 16S sequencing and now metagenomic sequencing and asked, what do you see in different places? And if you go to like the Nature or Science or the flagship publication that, you know, that you see of all this stuff, what often you see is you see graphs like this, where people try to present to you collectively all the data that they've collected and ask, what do you see? So in the Human Microbiome Project, for example, what people said was that if I look at the leftmost figure, what they did was they looked at different phyla. So each color here is a different phyla. Each column is a different individual. And they asked about the relative abundance of different phyla across individuals for different body slides. So this is the plaque on your mouth, this is your tongue, this is stool. And what the basic argument was that you can see that first, that there's quite a bit of variability across people, you know, at a given body site, but there's actually much more variability across body sites than there is across people, right? So the tongue of two different columns here looks much more similar than the plaque and the tongue of the same person, which is not really that surprising, because we would say, well, that's obviously true because the tongue environment is the same across people, much more similar than the tongue and the plaque environment. But you can do all these kind of things, and you could basically do, say, a PCA plot where you take different body sites, you color them, and what you see is indeed what you see by eye, which is that bacterial communities seem to basically indicate not what person you're at, but what body site you are. So they seem to select for environment. You can zoom in even within a single environment, you get some characteristic patterns, and these patterns seem to occur across everywhere, right? So these large-scale surveys have basically revealed this basic idea that you know, the environment seems to be dominating the human microbiome. So after the human microbiome, you know, there was this idea that, okay, we're too human-centric in the microbial world. So let's take a survey of all bacterial communities across the Earth. And this was called the Earth Microbiome Project. And when they published this about 2-3 years ago, they were looking for big scale patterns. And the patterns, they basically, you know, the two major things that they wanted to highlight in their publication, they said, what do we learn from all this stuff? What they learned was that it seemed that if you look at the microbial diversity, which is basically the y-axis here, as a function of, say, environmental harshness. So one proxy for environmental harshness is pH. Another thing for environmental harshness is temperature. It seemed that, you know, unsurprisingly, diversity seems to be peaked in environments that isn't that harsh, right? So the harsher the environment, too acidic, too basic, too high a temperature, too low a temperature, you don't get that much diversity, which is, again, it is what it is. You can think it's surprising or not surprising, but that's what they found. Another thing they really highlighted that they wanted to understand is that they looked at all these environments. So across the bottom axis here is all samples sorted by richness, right? And what they've done is they've sorted these samples. So over here on the right-hand side of this bottom figure are the most rich communities that you could find. And on the left-hand side are like the harshest environment with the least diversity. Then the y-axis are basically the different phyla. So the different phyla of microbiome that occur in each environment. And if a phyla occurs in an environment, what you see is that they put a little black box there. And what you see is that, you know, one of the things they pointed out is that the phyla that occur in the least diverse environments also occur in the most diverse environments. So there's lots of phyla in the diverse environments that don't occur in environments with not much diversity, but everything that occurs in environments with not much diversity occurs also in the high diversity things, right? And so they call this nestedness. Nestedness is something in ecology people care a lot about. So this is another big pattern that people observed in the Earth microbiome. What's interesting is there's also reproducible patterns when you go back and try to do this kind of thing where you try to, like, say, instead of looking in the wild, can I take these organisms and these environments and examine them in a more controlled lab setting? So Josh along with Alvaro, and I designed these experiments, which I didn't do any of. Josh and Alvaro and Nancy, a postdoc in Alvaro's lab, did all these experiments where we said, okay, can we now, instead of having really wild environments and really wild communities, can we at least ask what happens when we take really wild communities and grow them in really well-defined environments? And so what we did is we went out and got, wild communities and then grew them in these kind of what we call minimal synthetic environments. So what we did is we take like M9 media, which is like just the simplest media you can have, put some glucose in it or a single carbon source, and then serially passage these things and ask what survives, right? And so you basically just grow these up, dilute it, grow these up, dilute them, and this shows you, the dilution round, this shows you the relative abundance, and what happens is the community converges to some taxonomical composition. And the interesting thing we found is that this taxonomical composition could vary at the level of environmental sequence variance. So if you want to say individual species or some proxy for species, but at the family level, it seemed to be pretty consistent, right? And this was true independent of the starting community. So we started from very different communities and things seemed to converge to the same family level community. And this is shown on the right-hand side of this slide, which shows that we did this for 3 different sugar sources: glucose, citrate, and leucine, or carbon sources. And what we could do is we grew a bunch of communities, 12, from 12 different starting points. And this, kind of simplex shows you the family level abundance. So the communities seem to be dominated by 2 families, these enterobacteria and the pseudomonads, and there were a bunch of other families. And you could color the resulting communities by either the environment they were grown in, which is this red, blue, or green, or the starting community that they came from. And what you see again is that you get a lot of structure based on what environment they're grown in, but you really don't see any structure if I color them from their initial community. And so again, this suggests that the environment is basically the thing that's driving this community formation. So we're in these kind of selection-driven regimes, right? In going back to the fiber. And you can keep on going with other lab experiments. I won't show you. So these are some lab experiments from Otto Cordero's group, where what they did was they took these kind of ocean communities, they grew them on these kind of like beads with a single carbon source. And what they did is they sequenced these things in time and followed different taxa. And you saw this kind of very interesting pattern where you kind of had certain taxas that colonized these beads, but they kind of got wiped out until you got this final community that's dominated by these taxas down here. And so this is what in ecology is called succession. So they saw these kind of ecological succession. And you could do this again with different carbon sources. In all of them, you saw succession. And you got even more stuff like if I, instead of growing them on a single carbon source, I grew them on combinations of carbon sources and asked what happens. It turns out that you can predict the final community on the combination by just taking a linear combination of the communities on each of the individual things, each of the individual carbon sources. So again, this indicates that what's driving all this it doesn't seem to be really what's colonizing, but the environment, right? And from basic ecological theory, this suggests that the dominant thing that seems to be shaping all these microbial community is ecological selection. And so the goal of my talk is to basically show you that these kind of patterns, these are just a few of them. We have a huge list of basically every experiment and every pattern that we could find in the literature. And we want to know, is there a simple origin for all these patterns? And if we have a simple origin, maybe this will allow us to identify the relevant ecological and biological processes that are dominating all this stuff. So the 3 major claims of the talk, again, the first one we've already hit on, so I won't dwell on in the primer, which is that these classic, even though this seems to be, all these patterns seem to be driven by selection, the classical ecological models are going to be still inadequate for understanding microbial systems. So we have to generalize niche theory to account for all this kind of cross-feeding that we see in these things. The second thing that I'm going to show you is that actually all these patterns that I've shown you, I can do by ignoring all the details and instead just like kind of treating these things like typical random ecosystems, we call them, where we basically say, forget all the structure, let's replace all these details with random matrices. And what I'm going to show you is that just doing this very stupid thing where we basically take a classical ecological model, modify it to include cross-feeding, and then replace all the detailed structure that you get from all the genomes and things with random matrices, you can actually reproduce all these patterns. And very generically, robustly, and easily. And then the final part of the talk is I'm going to show you why we think that most macroscopic properties of healthy, diverse communities will almost always, you can predict them using these random ecosystems. In fact, I would argue what defines a healthy microbiome is precisely that it's indistinguishable from a random ecosystem. And that disease is very much a symptom of not being typical. It's an atypical state. So that's essentially what's going on. And that's what I want to say. And so, as I said before, traditional niche theories, are about competitive exclusion. You basically have niches, species fill these niches, and the number of surviving species is strictly limited by the number of limiting resources in the environment. And again, as we've highlighted in the model, the main problem with this is that it's really hard to separate out what resources are going to be present in the environment from what microbes are present in the environment, because microbes are continuously consuming these small molecules and producing these small molecules. And so cross-feeding means microbes construct their own environment. So that's one intuition we already discussed. But the second general thing I want to point out is this kind of very classic paper that defined all of condensed matter physics, which is this kind of paper that I urge everyone to read if they've never read, from 1972 by Phil Anderson, called More is Different. And the basic argument Phil Anderson makes is that intuitions from small systems often don't scale up to large systems, right? And so the basic idea is that intuition from few species, our claim is, do not scale up to complex ecosystems with many species because you get the emergence of new collective behaviors that aren't present in these small ecosystems, the kind of ecosystems people like to study in the lab. And it's like, I like this quote, the rule of talks is that if you're going to put a block quote, you have to read it out loud, so I'll read it out loud for you, which is this quote from Phil Anderson, which says that the constructionist hypothesis breaks down when confronted with the twin difficulties of scale and complexity. The behavior of large and complex aggregates of elementary particles, it turns out, is not to be understood in terms of a simple extrapolation of the properties of a few particles. Instead, at each level of complexity, entirely new properties appear. And this seems like something that should doom us, right? Like, how are we ever going to understand these complex communities if we can't understand the simple communities? But actually, in physics, we understand that there's basically three kinds of problems we can solve. We can solve one particle problems, two particle problems, and then we can solve infinite particle problems. But it's really hard to solve a problem with 10 particles. And the basic reason is that once you have enough things get complicated enough, instead of using kind of mechanistic intuition, we can use statistical intuition. And so what I'm going to show you is that we tried to implement this to try to understand microbial ecology. And the basic idea is to consider diverse typical communities that exchange metabolites and modify the environment. And we're going to give up on understanding any particular interaction and instead take a statistical viewpoint. And so the essential ingredients of our model is to basically take this consumer resource model that we talked about before and add to it basically metabolite exchange. So we're going to basically say what's going to happen is that we have consumers that consume individual resources, but they also, when they consume resources, produce new metabolites. So there's these kind of new exchange that's put in. So that's one thing, that's the selection part. The second thing we need is we need to worry about dispersal. So what we're going to imagine is that we have stochastic colonizations of environments. So you have some regional pool, and from this regional pool, you subsample different species to get different communities. And finally, what we need to do is say that each environment is different. So different environments are characterized by different resources being supplied to that environment. So if we do all that, essentially, we can write down a very, very complicated set of differential equations that we're going to use to modify these things. And the basic differential equations are basically summarized in these figures here, which are that, again, what I'm going to have is that I'm going to have species, and these species basically grow at some rate proportional to the input flux of all resources that come in. So I have some resources R alpha, they're consumed at some rate Ci alpha, so species I consumes resource alpha at alpha, and so I have an input flux J in I alpha, and it grows proportional to the input flux, minus some minimum energy I need to maintain myself at steady state. What's really important after that is each resource is supplied at some rate, RO alpha, but there's also exchange. It's also produced through this kind of metabolic exchange. So you have input, things go into the cell, things come out of the cell, and basically things come out of the cell. What comes out based on what's in is based on this stoichiometric metabolic matrix, which we'll call D throughout the talk. So basically, metabolism in this way, all of ecology in this way, is basically defined by two matrices. A consumer matrix that tells you what different bacteria like to eat, and a metabolic matrix that tells you that if I eat molecule alpha, how much of molecule beta do I produce? So the real question is, how do we choose all these parameters? Because I told you we want to think about species, hundreds of species, thousands of small molecules. And the key idea, again, as we've emphasized,
Pankaj,
is that,
one quick question, Pankaj.
Yep.
From the chat here, to take a statistical mechanics view, you have to assume that the system is ergodic and well mixed, right? Why is it true for an ecological system?
Okay, so I would say that's a misreading of statistical mechanics. Statistical mechanics doesn't actually assume ergodicity, it assumes typicality. And the difference is that, maybe we can come back to the discussion, but the claim is, I would claim that the new thing we've learned, and Luca, who's on the call, is actually one of the people who's thought about this a lot in the context of quantum mechanics, is that one of the things we've learned is that you don't actually need ergodicity. What you need to assume is that something is typical. And by typical, it's like, it's already there in information theory. It's what's called the AEP property in Shannon. But if I toss a coin 10,000 times, I only have to worry about configurations that have 5,000 heads, plus or minus something. And ergodicity is one way to get typicality, but it's not the only way to get typicality. And I'll give you an example. Like, so for example, the first place where this kind of approach was used was by Wigner to model the uranium nucleus atom. And what he said is that, oh, this Hamiltonian is too complicated, I'm just gonna replace it by a random Hermitian matrix. And there's no ergodicity. All it's saying is that uranium is not that different from any other very complicated metal, complicated atom with lots and lots of shells. And so that's my answer. My answer is that these kind of things rely on typicality and not ergodicity. And that actually someone needs to write a proper StatMech book that emphasizes typicality instead of ergodicity as the root of why StatMech works.
It's like very high probability ... right?
Yeah, exactly. Yeah.
That's all it is.
Yeah. So how do we choose all these parameters? Again, the idea is that when things get large, when I have a large number, I only have to worry about typical configurations. This is why information theory works. This is why everything works, right? This is the AEP principle. This is why StatMech works. And the price you pay, of course, is that if I only think about typical configurations, I can only talk about macroscopic quantities, things that are averages, right? Like pressure in a gas or temperature. But I can't tell you where any particular molecule in a gas is going to be, ever. Thermodynamics just has nothing to say about that. StatMech has nothing to say about that. And so, as I pointed out, this idea of typicality was first, raised by Wigner and Dyson in the context of uranium. And the basic idea is that in complex systems are typical and hence can be described by random matrices. And this actually has some tradition in ecology through a series of kind of, I would argue, very famous but kind of misunderstood papers by Robert May that he came out with 1972, where he wrote something about stability and complexity of ecosystems. And so the idea is that what we're going to say is that most microbiomes are typical. And if most microbiomes are typical, then I can replace these kind of matrices, these consumer matrices and these metabolic matrices, with random matrices. all right? And random matrices can have different amount of structure in them. All random matrices aren't the same, right? And so what I can do is by drawing random matrices from different ensembles, I can control how much structure I want to incorporate into my models. So for example, one thing I can do is just draw these kind of consumer matrices and these D matrices in an unstructured way from some kind of random matrix. Or alternatively, what I can say is that metabolism is really about different kind of tiers of molecules. So I have like different classes of molecules, sugars, carboxylic acids, amino acids, so on. And different species specialize, are better at eating certain things than others. In that case, I'll draw these random matrices from in some block structured way, right? And the claim is just by using these kind of either block structured random matrices or just uniform block structured matrices, actually we've been able to reproduce every pattern we've seen in every experiment and in every survey. And it's kind of surprising and with very minimal assumptions. And so,
what kind of patterns do you mean? So like, I mean, with the classic Wigner-Dyson thing is about the spacings between the eigenvalues, for example. Absolutely. So what kind of patterns?
I'll show you. So it's all in the scientific report, but basically all the patterns I showed you before, all the stuff all the kind of patterns that we seem to be able to measure with crude 16S sequences seem to only reflect this very coarse-grained taxonomical stuff. That's what I would say. Any kind of pattern that is basically, here is some thing where we measured the 16S pattern in some environment, right? How much metabolic detail are we putting in? I'm saying we're not probing much more than the fact that there seem to be families that consume stuff. And in fact, I can tell you exactly what level of coarse graining I need, because there's most of the patterns I can even reproduce with this very simple thing where I don't even put in resource families or species families. And then for other patterns, I have to basically put in a little bit more block diagonal structure. So what I like about this approach is it allows you to interrogate exactly what assumptions you need to reproduce the pattern that you observed with 16S.
I'm just wondering how you're using the word patterns. Like what do you have in mind?
I just mean the kind of stuff I showed you before. So I'll show you a bunch of examples, but like these nestedness, these PCAs, like just the kind of things I was showing you at the beginning of the talk.
Okay.
Anything, basically any figure you see in a paper that has 16S, this Otto Cordero thing, the things we saw in the science paper. So I'll show you, that's the whole next part of this.
So you're just basically arguing that these are very generic universal structures that emerge.
Yeah, exactly. That really, they're not probing much biology.
Right.
But if you read the papers, it seems to be a story about very particular biology.
Yeah.
So and we can make of it what we want. We can discuss whether we can do these things. So I should just say, if you want to play around with these kind of things, we wrote a pretty straightforward and easy to use Python package called the Community Simulator that allows you to play all this stuff. If you go to our GitHub site, Emergent Behaviors in Biology, we have all the code to reproduce all the figures I'm going to show you, and you'll see that we're not really cheating, and it's pretty straightforward, right? The final thing I should tell you is that it's actually a little bit hard to run these simulations, because you can imagine if I have thousands of species and thousands of resources, that these differential equations are really, really stiff, and they're hard to converge. But one of the things that we figured out in the last 2 years is that actually these kind of, the steady state dynamics of these kind of models are actually dual to constrained optimization problems. And so you can actually use constrained optimization combined with some kind of expectation maximization algorithms to quickly calculate steady states. And the Python package already has that implemented in it. And that's why we can actually test some of these ideas, because without this kind of like technical tool of the mapping to constrained optimization, we wouldn't be able to, it would just be too hard and too noisy to integrate these equations. So there is always a technical trick that allows you to do stuff. I didn't feel like talking about it, but it's basically laid out in these 2 papers here, and it's already implemented in this simulator. So as I said, what kind of patterns are we talking about? So this has largely been published in this scientific reports paper and in this science paper with Josh. So, for example, here is a pattern that was highlighted in the EMB, right? Which was this idea that you got nestedness, right? That the taxa that appear in the least diverse communities also, also appear in the most diverse communities. And all we said was that imagine what defines an environment is how harsh it is. And that's basically corresponds to changing this kind of MI, which is like the death rate of a bacteria. And if we just randomly sample that and use this kind of very simple block structure, you can actually just get this out for free. All right. So it just seems to be that any community where you have stochastic colonization and you have some fluctuation in harshness, you're going to get this nested structure. It doesn't seem to reflect anything except for the fact that selection is dominating stuff. So how do we know that? So it's not good enough to just produce a pattern. You better tell me when your model breaks, right? To learn something, I can't just reproduce a pattern. I have to tell you when I don't reproduce the pattern. And so you can ask, when do you not reproduce the pattern?
Pankaj, real quick.
Yep.
I wonder if you could say just a little bit. I mean, this is striking, the top plot, bottom plot, looking the same. So you have randomly generated these MIs here, the death rates.
Yeah, so what I've done is I've just taken a regional species pool, right? And I've just randomly sampled into different environments, and each environment has a different MI.
Yep, okay.
Good?
Okay.
And I've just block matrices for C and block matrices for D. All right? And you get this kind of thing. Now we wanted to know, I mean, so we had this really good referee for this paper because we were done, we felt very proud of ourselves. And the referee was like, okay, you can reproduce it, but tell me when you can't reproduce it. That's how we'll learn something. And so actually we had to work very hard to figure out when you couldn't reproduce this because this seems to be such a generic pattern. That's probably why we observe it. Well, it turns out that if what's limiting, you know, what species are present in a community isn't selection, isn't actually the dynamics at a place, but actually dispersal. So which bacteria end up in a given community to begin with, you lose this pattern, right? So this allows us to say that this is a signature of selection and not dispersal. So this is the kind of thing I think you can do with these kind of models, which is kind of interesting, right? So again, you know, you take the Earth microbiome project, the human microbiome project, and we saw that, you know, different body sites had similar things. You could PCA across body sites. There was some kind of fine-grained use structure, which is actually doesn't even require anything. That's true of random data. It doesn't matter. You can do all this stuff, and you can reproduce it. And again, what this is saying is that what we changed is that we just said that you have different resources present in the different body sites. So as long as you just have slightly different resources at different body sites, you're going to get this for free, right? But what's really important, again, to break this thing is that what you need is that you need the environments to be simple enough. Basically meaning that the environments should be dominated by a few resources, externally supplied resources. If you make the environments really complicated where you'll supply very, very many resources randomly, then you kind of lose this clustering. So we would argue that what these models suggest is that you can reproduce these patterns pretty easily, but what they're really telling us is that these body sites seem to be dominated by a few dominant resources that are setting up the whole cascade of cross-feeding. If you're not dominated by these few resources, you can't reproduce this pattern.
And I guess just linking it back to random matrix theory.
Yep.
You relate this to the distribution of eigenvalues and separation between.
Yeah, yes. So I have 5 minutes. The last 5 minutes is all going to be about why this works. But no, this is basically saying, actually, I'm not changing the random matrices that I draw here. All I'm changing is what I'm changing is the actual environments that I grow these things in. So here I'm changing the externally supplied environment here. So again, you have different things you can play with. Remember in the primer I said there's the environment you set up things in, there's dispersal, and there's selection. And so what we're trying to do is we're trying to isolate which of these forces are driving these kind of things. And so what seems to be driving this is basically the externally supplied resource. And that's what this is saying. It's saying that the external environment is very different here, which is not kind of surprising, though plaque and tongue, you could argue they have the very similar external environments, but they don't. And if you make the environments too complicated, you don't get these. And I'll tell you why this works with random matrix theory in a second. So this is just to show you that this is this paper from Otto. We can get that for free. We get succession for free. We get this modularity for free. This is the science paper that Josh set up. We can get all this kind of crazy thing for free from random matrix theory. So all these kind of things, everything we've measured, really, and I'm not being facetious, everything that looks like it's a generic pattern that we try to reproduce, we can, and with very little manipulation. Of course, we have to think about what exactly is the environment is doing. Are we limited by dispersal or not dispersal? But once we set that up, we can do these.
And to what extent do you, when you say reproduce, do you mean qualitatively or even quantitatively? Are there laws that you've looked at quantitatively, or?
I don't know what it means to do these things quantitatively, because I can fiddle with the random matrices how long I want. Yeah, we can discuss about what the value, what p-values is.
So the Wigner-Dyson-Mehta thing is quantitative as well, right? It's about the law of the spacings. So
the law of the spacing, but prime numbers also show that law. So it's unclear what if I go to the Riemann zeta function and I look at the spacing.
Yeah, you get the sine law too, sure, but
so I'm not sure that's a probe. That's not a probe of anything to do with uranium in the same way.
Okay.
That's my claim. My claim is nothing to do with uranium. The Luca should be the one who's really saying things about this, because this I don't know. Luca knows more, but yeah, I would claim that's not a probe of uranium. That's a probe of.
Sure.
Huh?
No, I agree. I'm just wondering how universal.
No, that's the question. The real question is, like, so this paper ended up in Scientific Reports because I refused to do a p-value on principle. This is a constant fight between me and Josh too, because Josh is a computational biologist and I think p-value should be banned.
Okay.
Okay, so the last part, because I have 8 minutes, is but why do random metabolisms and random consumptions describe experiments so well? Like, so we got really confused because we really didn't think it would work that well. That's the honest truth. Like, I thought, oh, it'll do okay. But we had enough what I would call human out of sample tests, where we wouldn't like set it up to cheat and someone would show us an experiment after we wrote the theory and it would still reproduce it with almost no fidgeting, that we were surprised. And so this is the strongest claim of the talk and the most technical claim, which is that we think that macroscopic properties of healthy, diverse communities will almost always behave like random ecosystems. That what defines a healthy, diverse community is that it's typical. And so to better understand this, we wanted to, like, you know, and this is going to be the mathy part of the talk for the last 10 minutes. So you'll forgive me for doing some math, because I am a theoretical physicist. I try to avoid it, but I can't help it.
We don't apologize for math at MIA.
Okay. Well, so this is the math. So what we wanted to understand is how can we understand these kind of things better? Like what's going on? So we set up this kind of like thought experiment. So imagine I take some consumer resource model and I engineer it. So I engineer the interactions in some very particular way. So the simplest engineered interaction you can imagine is that I make a community of specialists. So this species eats, you know, diamonds. This species eats circles. This species eats, you know, stars, and so on. And now I ask, imagine just by chance, I have some off-target interactions, right? And so the basic idea is I have some structured matrix B, and to that I add some random component C, all right? And then I run it through these differential equations. So I have these dynamical equations. This is a particular model we analyzed, which is the original MacArthur consumer resource model, but it doesn't really, it's not really super important. And I ask, how do these, you know, how do the steady states and resulting steady states differ? And what we find is, what's interesting is that the parameter that we were going to ask is we're going to control the amount of noise that we add, right? So the amount of noise that we add is basically controlled by the standard deviation of this random matrix C that we add. And what you can ask now is you can plot some macroscopic quantity here. And it's not really important for important what it is. Here is just the fraction of species that survive in the ecosystem. So if I have no noise, it's clear every species can survive because they don't compete with each other. Right? This species just eats this resource, this species eats this resource. And as I add up the noise, as I add noise to the system, what's going to happen is these species are going to start competing with each other more and more. And so now I can ask, how does the fraction of species that survive change as I change this noise, sigma c? Okay? And what's really important for all this, and I want to point out, is that the way we scale this sigma c is that we scale it with the number of resources. Right? Sigma c over square root of m is the number of things that add up. And so what you can see is that, this is for m equals 100. What you can see is as I increase sigma c, basically this thing looks like it's not competing, and then suddenly, as I hit sigma c approximately 1, I'm indistinguishable from a completely random matrix.
Sorry,
So yeah.
What's your order parameter?
The order parameter is just the fraction of species that are surviving, but I could do this for the mean abundance, I could do it for the variance, I can do it for any quantity that is a sum of many, many things. So this is just the number of species that survive in the ecosystem.
Okay.
Because they start competing with each other more, right? So some of the species are going to go extinct by competitive exclusion.
What's blue versus red?
Blue is the structured matrix, and red is the completely random matrix without this kind of diagonal. So you see there's a diagonal band here.
Oh, without the diagonal. Okay.
Right? And the point is that when I add some 10 equals to 1 noise, order 1 noise, basically, even though this matrix looks like it's essentially the identity matrix with some noise, and we can make the noise, and I'll show you, and this is completely unstructured, the actual macroscopic properties are completely indistinguishable from each other. And the bigger I make the system, oh, I don't have the slide, the more dramatic this effect is. Because remember, the actual noise scales like 1 over square root of m. So if I have 1,000, this phase transition still exists, it happens at sigma c equals 1, but the effective amount of noise is much, much less. So even though these matrices look very structured, the actual macroscopic properties are indistinguishable from a completely random matrix. And you can do this with the identity matrix, you can do this with different kinds of noise, you can do this for different kinds of engineered things, and you always see this kind of phase transition to typicality. And so the question is why? So this suggests that even adding a small amount of noise, off-target effects, even if I take a very structured matrix, you're going to get this phase transition where the actual results of the dynamics look indistinguishable if I look at some macroscopic quantity from a completely random ecosystem. And so how do we understand that? The way we can understand that is we have two different approaches for understanding these. One is a random matrix theory approach, and another is an approach based on something called the cavity method, which is a mean field theory approach from disordered systems in physics. And so the basic idea of this kind of random matrix theory approach is that I take this original consumer resource model, and what I can do is I can set dr alpha dt equal to 0, solve for r alpha, and plug that back into this top equation here. If I do that, you'll see that what I get is this kind of generalized Lotka-Volterra model, where the interaction matrix Aij is exactly this Wishart matrix, which says how much do species compete for the same thing, right? So it's basically the overlap of the consumer preferences. The more they overlap, the more they compete, the bigger alpha Aij is. So what I can do is I can just show you how this Aij looks at different sigma c's. And what I can see, what I know is that if these c's were completely random, the statistical properties of these Aij's would be described by what's called the Marchenko-Pasteur distribution, because that's what a Wishart matrix describes. And so if I do this, you can see that if I fit the spectrum of this Aij, at these kind of points here, it looks very different from Marchenko-Pasteur, but suddenly, as I increase the noise a little bit, it's very well described by a Marchenko-Pasteur distribution, and you can show it's exact where it's going on, right? And so the random matrix theory can be characterized by the spectrum of these kind of effective interaction matrices. Turns out it's really a generalized susceptibility. It doesn't really matter. There's many technical things I can tell you about these things. And it's essentially related to something that maybe some of you have heard of, which is this kind of information plus noise transition in electrical engineering. So there's this kind of well-established theory over the last 15 years in engineering, which says that imagine I take some structured matrix, I add some noise to it. This is called the information plus noise model. The question is, when can I reconstruct back the information, the structured information? And turns out there's a strict phase transition as a function of the amount of noise where you can't reconstruct that back. So this is called the information plus noise model. And this corresponds to precisely a point where some minimum eigenvalue hits 0 in this kind of matrix. And you can go through and you can show that that's exactly what's happening here. It's kind of like this information plus noise transition in signal reconstruction. And there's another approach on spin glass theory, which is based on the cavity method, which I won't go through, but it basically gives you the same exact thing. So that's what I wanted to say, basically. So what I wanted to tell you, the major points of the talk were that classical ecological models are inadequate for understanding microbial ecosystems. These kind of large-scale reproducible patterns that seem to be probed with 16S sequences across lab and natural environments seem to be able to be produced with very stupid assumptions, right? I take a classical ecological model, add crossfeeding, and then draw random matrices with very minimal structure. And by doing that, I can probe what ecological processes are going on. And then we kind of have this idea that if I take a species, if I take a microbiome and I add noise to it, like even though there might be a lot of structure by eye that I see, because you have this information plus noise transition, you'll almost always behave like these random ecosystems, right? So, and so I'll add to that what I think disease is exactly this idea that this kind of typicality breaks down. And for those who are interested technically in what's going on, it's basically the idea that I can add one more species, and instead of having a 1 over n, 1 over the number of species effect on the environment, it actually has a macroscopic effect on the environment. So when I can add one species and it has a macroscopic effect on the whole environment, then all this kind of typicality arguments break down. And that's exactly what disease corresponds to. And I think you can make a similar argument about genomics, that if I flip one gene and I make a macroscopic change on the whole gene expression pattern, then typicality arguments break down, and that's kind of a signature of disease. But anyway, that's all speculation. So let me end and say these are the people that this work was really done by it's really a collaborative effort of Wenping, Bobby, and me, and Josh for the science paper, which I didn't largely talk about, but is really the foundation for a lot of stuff, and the NIH and the Simons Foundation for giving me money to babble about all this nonsense, so thank you so much for listening.